4 more new strands will be separated. 4 new strands will be synthesized. And now they will fuse with the parental strands. And now how they look like, we will get 2 hybrid strands and 2 lighter bands, right? So 2 lighter bands and 2 hybrid bands. So like this we'll get. So upon this experiment, they concluded that the DNA replicates according to the semi-conservative mode of replication. Because if it is either conservative or dispersive mode, you may get different results. Okay? Is it clear? So you can just practice by adding the labels, so it will be more clear if you don't understand. So this is how the experiment looks like. So here, first E. coli was grown in the presence of N15 heavy isotope, and then they have transferred to the N14 light isotope. Then again, the cells were broken and they extracted DNA, and they added this DNA into cesium chloride solution and they centrifuged. Upon centrifugation, so the bottom, if the both strands are heavy strand, you will get a band at the bottom. If they are hybrid, you will get at the top center. And if there are 2 light strands and that is 2 hybrid and 2 light strands, you will get 1 band at the top and 1 band at the center. Okay. So this is the same thing. Then, so the next is 3 rounds of replication. Okay. When they performed, so half of the parental DNA molecule is conserved in each new double helix, and the newly synthesized complementary strands will be half. So it is semi-conservative. So 1 strand is from the parent and 1 is newly synthesized strand. And it is bidirectional. So why it is bidirectional? Because when the 2 strands get separated, so from the origin of replication, especially in bacteria and prokaryotes, we will have origin of replication, from where the replication starts. So it is not in only one direction it is taking place, but it is taking place in the 2 directions simultaneously. Therefore, it is called as bidirectional replication. So the replication starts from the unwinding of the double-stranded DNA at a particular point called origin of replication. Then the synthesis takes place. Now what happens? This parental double-stranded DNA forms a Y-shaped fork, replication fork. That means this is also called replication bubble. That means when the 2 get separated, so in this image you can see, so on the either side it is not, it is together, but only at the center the 2 strands got separated. So it is looking like a bubble, or it is also like if you just cut it at the center, it looks like a Y shape on the both sides. So therefore it is called replication fork, okay, or Y-shaped structure. So this is how it looks like. So 1 strand is from 3' to 5' and other strand is from 5' to 3'. So the 3' to 5' is called as leading strand and 5' to 3' is called lagging strand. Why? Because always the DNA replication takes place in the direction of 5' to 3'. So when it is going from 5' to 3', the template strand should be in the opposite directions because they are anti-parallel. So opposite direction means the template should be in 3' to 5' direction. Right? So it will continuously takes place in that strand. So therefore it is called as leading strand, where the continuous DNA replication takes place. Whereas in the other opposite strand, it is called lagging strand because a discontinuous mode of replication takes place. Because now as the template strand is from 5' to 3', now the replication has to take place from 3' to 5'. But that is not the case. Always DNA replication takes place only in the direction of 5' to 3'. So this is opposite way. So therefore it cannot continuously synthesize the strands. So therefore it is called as lagging strand. And in prokaryotes, they form a theta structure. Therefore, it is called theta replication because the replication process starts from the origin and proceeds into two opposite directions. Therefore, it is called theta replication. So here you can see in the image how the replication is taking place. Now there are various enzymes and proteins which are involved in the synthesis of DNA replication. One is RNA polymerase. So this polymerase is important for joining the correct nucleotides to the adjacent nucleotides. And second is helicase. Helicase is involved in unbinding the DNA and melts it. Okay. So first step itself is unwinding. So helicase is the first enzyme which comes and binds and it will separate the replication that is separated DNA strands. Then primase. Primase forms RNA primer. Okay, to start the polymerization. Then another is single strand binding protein. So when the two strands get separated, they should be stabilized. They should not wound again to each other. So therefore, to stabilize that, single stranded binding proteins comes and binds. Then gyrase. So gyrase is a topoisomerase. Topoisomerase is nothing but in the previous class I told you about supercoiling. That means because it is helical in nature, it will try to coil itself around itself. Okay. So to remove that strain, gyrase enzyme helps in relieving that strain. Then ligase. Ligase is for joining the strands. And telomerase. So it is to finish the ends of the DNA strands in eukaryotes. Now we'll look into the DNA replication. So enzymes, so other proteins which are involved are DNA A protein. First, this is the protein which will recognize the origin of replication. Then DNA B protein is also called as DNA helicase. I told you it will open the double strand DNA. Then DNA C protein, so which assists the DNA B to bind to origin of replication. Then DNA polymerases, which synthesize the new DNA strand. Then DNA G protein, which is DNA primase, which synthesizes RNA primer. Single stranded binding protein, so which binds the single strands of DNA. DNA gyrase, which will remove the strain. So first we look into the DNA polymerases of prokaryotes. So first is DNA polymerase 1. So this is called as DNA-dependent DNA polymerase, which was discovered in 1958 by Arthur Kornberg. He got Nobel Prize for this. Hence, this enzyme is called Kornberg enzyme. Later, DNA polymerase 2 and polymerase 3 were also identified. So this polymerase has two activities. One is 5' to 3' polymerase activity. That means they synthesize the DNA strand from 5' to 3' direction. And it also has exonuclease activity. So exo means from outside. So if any misincorporation of bases are there, so this exonuclease comes and removes the nucleotides from the 3' direction to 5' direction. That is from the end. Okay, so that it will remove all the nucleotides until where the mismatch has happened. And from there again the synthesis takes place. So the exonuclease functions are 5' to 3' exonuclease activity and 3' to 5' exonuclease activity. So usually all these three enzymes have 3' to 5' exonuclease activity. That means in the reverse direction, it will remove the nucleotides whenever there is any mismatched nucleotides are present. But only one enzyme, DNA polymerase 3, I think. So it has 5' to 3' exonuclease activity. That means 5' to 3', we have a small segment of primer. So primer is involved in the initiation of DNA replication. So because the DNA replication cannot start by itself. So this primer has to be removed. So that segment has to be removed by this exonuclease activity. That is 5' to 3' exonuclease activity. Now this is the polymerase 1. So it is mainly responsible for proofreading and filling the gaps and repairing damaged DNA. So this DNA polymerase 1 has two fragments. One is small fragment and there is large fragment. So the small fragment has 5' to 3' exonuclease activity, and the large fragment, which is called Klenow fragment, having DNA polymerization and 3' to 5' exonuclease activity. So sorry, previously I told that DNA polymerase 3 has 5' to 3' exonuclease, but not on DNA polymerase 1 only will be having the 5' to 3' exonuclease activity. So there are two fragments, smaller fragment and larger fragment, and each fragment is having a specific function. Then polymerase 2. So this is temporarily functional when DNA polymerase 1 and 3 are not functional. So this is mainly for the synthesis of the damaged template. So it will have the exonuclease activity. So it participates in the DNA repair process. Then polymerase 3, so it's a heterodimer enzyme. So it has highest polymerization activity, and it is the true enzyme which is responsible for the elongation process. So it is the actual enzyme which is involved in the replication. So this is how the structure of polymerase 3 looks like. So it has different subunits. So it has two alpha subunits, which have 5' to 3' polymerizing activity. Then two epsilon subunits, which have 3' to 5' exonuclease activity, and they ensure the replication fidelity. So that is specificity and how accurately the replication is taking place. All these will be checked. And the theta is there are two theta subunits which maintain the heterodimer structure. And we also have beta. So here you can see in the image. Sorry. So these beta subunits are called beta clams. Okay. And the two two subunits are identical only unless, like, if there is one complex which is made up of delta, delta prime, psi and phi. So this subunit makes one subunit more heavier than the other. Okay. So here you can see. So this is the core where alpha, epsilon and theta are present, and there are two beta clams. So beta clams will act as a hook for fixing the two strands simultaneously. That means at the center you have this DNA polymerase, and there are two hooks. One is hanging to the one strand and other is hanging to the other strand, and it will start the scanning process. And you have, for dimerization, you have tau subunits, okay, at the bottom, and we have DNSB helicase activity. The nucleotides are always added to the growing strand at the 3' end, that is the end at which the DNA strand has free OH group. Okay, so the 3' end you can see where free OH group is there, which will involve in the reaction process. So OH group on the 3' carbon of its terminal deoxyribose. The next is DNA G protein, which is primase. So primase is necessary for synthesis of primer, which is RNA molecule. So, and here this primase is able to synthesize primers using free NTPs as a substrate and single-stranded DNA as a template. So here the primers are the short RNA fragments, which is having several nucleotides long. So this, you can see here the blue fragment, which is RNA primer. So RNA primer is required for the synthesis of new DNA strand, that is for initiation of replication. Primers provide free OH group to react with the alpha phosphate atom of the DNTP so that it will form the phosphodiester bond. So this primase, DNA B, DNA C, and origin, they all form a complex which is called as primosome complex at the initiation phase. So this, what is this B? DNA B is the helicase. So helicase, it will open the double-stranded DNA by consuming the ATP. And the opening process is assisted by DNA A and DNA C proteins. Then once these two strands get separated, there is another protein called single-stranded binding protein, which will come and bind to these two DNA strands so that they remain open and they do not form the double helix again, and also to stabilize the DNA and protect the DNA from nucleases. That is, there will be DNases and all these within the cell. So to protect this DNA from those, so this single-stranded binding protein it plays an important role. Then DNA gyrase. So this DNA gyrase is involved in the relaxing the supercoil. So how it relaxes? See, I told you in the previous class, like just imagine a big rubber band. So when you keep it like that, so it will try to like spin over its own, like it will coil around its own. So it will form many coils. So if you cannot open, so rubber band we can open, okay, but if it is something like a big one, some rope or something, what we will do, we will cut it from one side so that it will easily get open and it will form a single strand. So in the same way here, when the DNA gets supercoiled, this DNA gyrase cuts the phosphodiester bond on both strands so that it will release the supercoil and reforms the phosphodiester bond once they are separated. So it can change double-stranded DNA into negative supercoiling state by consuming ATP. Then the ligase. So ligase is nothing but it is involved in the joining of the strands. See, whenever a RNA primase, okay, so primer or anything is cut, there will be space. So to join the space, so usually DNA ligase is used because during some processes, like as in the lagging strand, the DNA replication is discontinuous, right? So you will get small fragments. So all these fragments should get joined. So this is done by the ligase. So they connect to adjacent single-stranded DNA strands by joining the 3' OH of one strand and 5' phosphate of another strand. So it will seal the nick, or nick means gap, in the process of replication, repairing, recombination, and splicing. So what is replication fidelity? So replication fidelity is based on the principle of base pairing. So that means with how much accuracy. So in the previous classes we have discussed like A should always form a bond with the T and G should always form bond with the C. So that means the bonding has to be accurate. If there is any mismatch, then it can result in the errors and can cause some serious diseases like cancer. So this is called the accuracy with which the base pairing takes place is called replication fidelity. So enzymes use two mechanisms to ensure the replication fidelity. One is proofreading and real-time correction and base selection. Proofreading and correction means the DNA polymerase 1 will find the mismatched nucleotides, it identifies and removes it by the 3' to 5' exonuclease activity. And again, it will add the correct nucleotides. Okay. Now we discuss about, these are the basics of DNA replication. Now we discuss the actual process of DNA replication. So it has three stages: initiation, elongation, and termination. So the three stages of replication are initiation, where it starts at the origin of replication. So separation of double-stranded DNA and primer synthesis. And in the elongation, it involves the addition of new nucleotides based on their complementarity to the template strand, and it forms the phosphodiester bond. Okay, if there is any mismatch also, it will try to repair everything. And then termination, where it stops the DNA replication at termination site. So this is bacterial DNA, where you will have origin ORIC, which is the origin of replication. So from where the replication starts. So the initiation, the replication starts from a particular point called origin of replication. So it will have a short segment of nucleotides, which is called 9-mer and 13-mer. Okay, so first the binding of DNA A protein comes and binds to this 9-mer. Okay, once it comes to and binds to the 9-mer, it will fold. This DNA A protein will fold this DNA, and by using ATP, and it will form a complex. This is called open complex. And to this, DNA B and C will come and bind. Now DNA B is helicase. Now this helicase will remove or separate the two strands of the DNA. And so the origin of replication is site where the DNA synthesis starts. Okay. So first DNA A recognizes ORIC, and then it recruits DNA B, which is helicase, and DNA C, which join the DNA and DNA A complex. It will separate the DNAs slowly, and once the DNA gets separated, single-stranded binding protein comes and binds and stabilizes this single-stranded DNA. Now, once it gets separated and stabilized, now the primase will join this other DNA C and B, other proteins, and forms a primosome complex, and it will start synthesizing the primer. So in the 5' to 3' direction. Now a short RNA fragment with free OH group, free 3' OH group, is ready for DNA elongation. Now this is about the initiation. And so these small fragments, so I told you in one strand it is continuous, in other strand it is discontinuous. So these small continuous bands on the lagging strand are called Okazaki fragments, because this was identified by Rizi Okazaki and Chuneko Okazaki. These were wife and husband, they are scientists. So because of their names, they got Okazaki fragments. Next comes elongation, where the D and TPs are continuously connected to the primers. And here the DNA polymerase 3 enzyme will start to synthesize. First, the core enzymes alpha, epsilon, and theta, they catalyze the synthesis of leading and lagging strand. And the nature of chain elongation is by formation of phosphodiester bonds. So this is how the bonding takes place. So here, so once the elongation is done, this primer, which is synthesized at the initial stage, so that is removed by the DNA polymerase 1, because it has 5' to 3' exonuclease activity. And in this Okazaki fragment also, we have small fragments, right? These small fragments has to be joined by the DNA ligase enzyme. So in the lagging strand synthesis, so the RNA primers on Okazaki fragments are digested by the enzyme RNase, and these gaps are filled by DNA polymerase 1 in the 5' to 3' direction, and the nick between the 5' end of the fragment and 3' end of the next fragment is sealed by the ligase. So now what are these Okazaki fragments? So these are the DNA fragments which are sequentially synthesized on the DNA template strand having the 5' end. And the DNA fragments are called Okazaki fragments, where they are 1000 to 2000 nucleotides long in prokaryotes, whereas only 100 to 150 nucleotides in eukaryotes. So the strand which is having these Okazaki fragments is called as lagging strand. So the directionality of the DNA strands. So here, so this lagging strand goes in the opposite direction of the fork movement, replication fork. But the leading strand will go in the same direction as that of the replication fork. Therefore, in one strand it is continuous and in another strand it is discontinuous replication. Hence, it is called semi-discontinuous replication. So this is the same. Now here you can see. Now initially, when the two strands are separated, this single-stranded binding proteins will come and bind. Then the RNA primer will form a RNA primase will form a primer, DNA primase. Then DNA B, that is helicase, comes and they separate. And now the DNA polymerase 2. So once the elongation is done, to prevent the supercoiling, the topoisomerase 2 or DNA gyrase enters and it will try to remove the supercoiling. Now, once the elongation is done, then primer is removed and the any gaps are there, they are filled by the ligase. Then the last step is termination. So the replication of E. coli is bidirectional from one origin and the two replication forks, and they meet at a point called TER, that is terminator site at 32 position. So all the primers will be removed and the fragments will be connected by the DNA polymerase 1 and ligase. So like this, we have 6 terminator sites, 3 are in clockwise and 3 are in anti-clockwise. So whenever this polymerase, it recognizes this terminator sequence, there will be termination process. So what are the inhibitors of this DNA replication? There are some chemicals like nalidixic acid, novobiosin, and ciprofloxacin. So these are the inhibitors of DNA replication. And adriamycin, etoposide, and doxorubicin. So these can be acted as antibiotics because they inhibit the replication process and they stop the infection. And this is about the prokaryotic replication. When it comes to the eukaryotic DNA replication, so it is closely related to the cell cycle because in the S phase of the cell cycle, DNA replication or DNA synthesis takes place. So this is how it looks like. So we have discussed a lot about the cell cycle. So I'm not going to discuss again. So in eukaryotes, we have multiple origins of replication on the chromosome. So they get activated in the sequential order rather than simultaneous. Okay, one after the other. Because it is a larger DNA when compared to prokaryotes. So here the DNA polymerases are DNA alpha, polymerase beta, polymerase gamma, polymerase delta, and polymerase epsilon. So polymerase alpha is involved in initiation of replication and synthesis of primers. Then polymerase beta is involved in replication with low fidelity. And polymerase delta is for mitochondrial DNA synthesis. Polymerase delta is for elongation, and epsilon is for lagging strand synthesis, proofreading, and gap filling. The first step here is also initiation. So the eukaryotic replication origins are shorter than the E. coli, and they consist of ARS, that is autonomously replicating sequences, or they are called replicators. So here this DNA polymerase alpha has primase activity and delta has polymerase activity and helicase activity. So here also same way, first The helicase comes, it will separate, and the primase will synthesize the small primers, and polymerase will extend. And polymerase delta requires a protein called proliferating cell nuclear antigen, that is PCNA. And it needs topoisomerase and replication factors also. This is additional to the prokaryotic replication. Now, once the initiation started, then the elongation, so where the DNA replication and nucleosome assembly occurs, and overall replication speed up is compatible with that of the prokaryotes. So the replication is almost same, elongation and initiation, everything, only some few more proteins are there. And here, once the elongation takes place by the polymerase, then comes whenever there are terminator sites, so there will be termination of this replication at terminator sites. Then in addition, in eukaryotes, we have a terminal structure called telomere, which is seen in the chromosomes. And this telomere is composed of terminal DNA sequence and protein. So they are rich in T and G nucleotides. So it is crucial for keeping the termini of the chromosome, because sometimes they will be lost, and they will, if they are becoming sticky, they will go and stick to each other, the two strands. So the eukaryotic cells use telomerase to maintain the integrity of the DNA telomerase. So it's composed of RNA, protein, and reverse transcriptase. So here they synthesize DNA using RNA as a template. Therefore, it is called as a reverse transcription process. So this is how it looks like. A primer is removed, so you will have a gap. So here, now as there is a gap, slowly using the template, synthesis will take place. Okay. By end, the gap is filled with the ligation. So these are the various proteins and their complementary proteins in the prokaryotes. So later you can just go through this chart. Now we have base analogs. So base analog is nothing but a chemical that is similar to the nucleotides. So we have purine analogs and pyrimidine analogs. So purine analogs, they look like the purines. Pyrimidine analogs look like pyrimidines. So mainly 5-fluoro or 5-iodouracil, 3-deoxyuridine, 6-thioguanine, 6-mercaptopurine, 5 or 6 azathioridine, 5 or 6 azacitidine, and 8 azaguanine. So these are all the what called base analogs, which are incorporated into the DNA prior to the cell division. So they can cause the mutations in the DNA. So these are the structures. So allopurinol, mercaptopurine, thioguanine, so all these. There are some other group of agents called intercalating agents. So these are the molecules that can insert between the bases of the DNA base pairs. Therefore, it can cause a mutation during replication. So ethidium bromide, proflavin, and donrobicin. So this is the structure of ethidium bromide. So these are the various intercalating agents. So proflavin and diaminoacridine. So these are acriflavin derivatives, and they have bacteriostatic effect against gram-positive bacteria. Then daunorubicin is commonly used in the treatment of leukemia, that is blood cancer. So acute myeloid leukemia, acute lymphatic leukemia, and also in the neuroblastoma. Then DNA polymerase inhibitor. So some also inhibit the enzymes like acyclovir, ganciclovir. So these two. So they are naturally occurring nitrogen base essential in DNA replication. So these are mainly for the inhibitors of viral DNA polymerase. So they act on like they used as a drugs against the viral infections. Epidiocolin, so which inhibits DNA polymerase epsilon in eukaryotes. Then DNA damage. So even though there are a lot of fidelity and accuracy is maintained, but still some DNA damage can occur under physiological conditions. And this damaged DNA is replaced with the normal DNA by the process called repair. So there are various physical and chemical agents that can cause DNA damage. There are endogenous agents and exogenous agents. So agents that damage DNA can be mutagenic, cytogenic, or both. So mutagenic means they can cause mutations and they can cause cancers like that also. And cytogenic, which will kill the or which will harm the cells. So DNA damaging agents that cause mutations are called as mutagens. So types of DNA damage are their physical, chemical, and environmental. So single base alteration means only in the one base there will be alteration like depurination, base alkylation, insertion, deletion, etc. Then two base alteration. So this is by UV light induced pyrimidine dimer. Okay, whenever we get exposed to UV light, pyrimidine dimers are formed. Then chain breaks. That means oxidative free radical formation and ionizing radiation. Exposure to ionizing radiation also can cause chain breaks. Then cross-linking. So between the bases of the same or opposite strands, and also between the DNA and protein molecules, also there will be cross-linking. So this is single base alteration. So if you change one base, so the cytosine can become uracil, 5-methyl cytosine become thymine, adenine become hypoxanthine, and guanine becomes xanthine. They are called derivatives. Okay. Now the DNA damaging agents. So we have spontaneous agents like high reactive oxygen radicals. So they produce the products during normal cellular respiration. And this ROS like hydrogen peroxide, superoxide, or hydroxyl radicals, they are very potent damaging agents. So this results in DNA damage causing like first the oxidation of nitrogen bases, deoxyribose, and strand breaks. So all these damages can be caused by the reactive oxygen species. So even the radiations also. So the high energy electromagnetic radiations to the exposure of which the cell experiences considerable change in their DNA. So one is ultraviolet rays. So we have three types A, B, and C, and the wavelengths are given. And then X-rays, gamma rays. So gamma rays, like through these direct damage takes place in the when DNA and water tightly bound to it absorb the radiation. So indirect damage takes place when water and other molecules surrounding the DNA absorbs this radiation and form respective species that form the damaged DNA. So here, because of UV, thymidine dimers will be formed. Then chemical agents like deaminating agents like sodium nitrate, sodium nitrite, then nitrosamine, nitrous acid, and then alkylating agents, dimethyl sulfate, dimethyl nitrosamine, and nitrogen mustard. So these are all can cause DNA damage by causing mutations. So mutation is a sudden change which occur in the DNA, and the chemicals which induces these mutations are called mutagens. So these can occur during replication, transcription, and translation, and they can also reflect on these three processes. Like if there is any change in the DNA, it causes change in the RNA, and which ultimately changes protein. So mutations are of two types: spontaneous mutations and induced mutations. So in the spontaneous mutations, normally it is not pre-planned, suddenly it happens. This is during DNA replication, whereas induced mutations are by treatment with the mutagens or chemicals. So then we have point mutation. That means only in the single base there will be mutation and the substitution takes place. Then these substitutions are two types: transitions, transversions. So in this case, in transitions, a purine or a pyrimidine is replaced by another. And that is, purine is replaced by purine, pyrimidine is replaced by pyrimidine. Then in transverse substitution, purine is replaced by pyrimidine and pyrimidine is replaced by purine. Okay. So this is a genetic code. Then point mutation. So in the genetic code, if you see, if one base is also changed, then the complete amino acid will change. So here you can see UCA is becoming UCU. So here, though the base change is there, the product remains the same. That is, both gives serine only. Then such type of mutation is called as silent mutation. And if UCA and ACA is there, so UCA gives serine and ACA gives threonine. This is called missense mutation. So the sense is changed. The nonsense mutation, so UGG is becoming UGA. UAU is becoming UAA. That means either tryptophan or tyrosine, the codons which are coding for these amino acids, because of change in one base, they are becoming stop codon. So hence the termination will happen prematurely. Then missense mutation, it is a point mutation that can change a codon so that a different protein is synthesized, which is non-synonymous change. So the best example is sickle cell anemia, okay, where one base is changed here. So because of this change, the complete protein is changed, and you will see the shape of the cell also changed. Then frame shift mutation, where one or more base pairs are inserted or deleted so that the codon, suppose you have 1, 2, 3, 4, 5, 6, 2 codons. If 3 is removed, third base. So what happens? Now 1 to 4 will become one codon, and 5, 6, 7 will become one codon. So that means the complete codon has changed. So the amino acid also will change, and you will get a different protein. Now, if this mutation takes place, so what are the DNA repair mechanisms that are available? So one is base excision repair, where the correction of the chemical damage is done, like uracil, xanthin, 3-methyladenine, formamidopyrimidine, 5,6-hydrated thymine. So all these are the chemical base changes where the chemical damage takes place. So here DNA polymerase 1 can perform base excision repair, and also DNA glycosylases, AP endonucleases, DNA ligases. So all these are involved in the base excision repair. So here these glycosylases, they are specific for very different types of base damages, and AP endonuclease recognizes the site with a missing base, and deoxyribose phosphodiester removes the sugar phosphate bond lacking the base. The nucleotide excision repair, where the cells are used to repair a bulky DNA damage. So it's non-specific. Okay. So whether it is UV photo products, or that is dimer formation or chemical adducts. So anything that can be removed by this nucleotide excision repair. So for pyrimidine dimers or any other dimers, etc. So here the examples are DNA polymerase 1 and ABC exonuclease. Then xeroderma pigmentosa. So this is a type of like defect in the nucleotide excision repair where, so this is by autosomal recessive disease where the patients are photosensitive and susceptible to skin cancer. So here there is a defect in the nucleotide excision repair. Okay. So therefore the mutations get accumulated and results in the disease. The next is mismatch repair. So those are the enzymes which are involved in the mismatch repair. So this mismatch repair system is an excision or resynthesis system. So it has 4 phases. One is recognition of mutation by mut-S protein. So first mut-S protein will recognize the mismatch. Then the repair enzymes comes and it repairs the site where the mismatch has happened. And the excision is or excision of the incorrect sequence till where this wrong base pairs are inserted. And again, by using DNA polymerase, the sequence is resynthesized. So this is how the synthesis. And there are some disorders which are associated with this DNA repair mechanism. So one is xeroderma pigmentosa. I told you it is autosomal recessive disorder. So genetic defects are, so these DNA repair mechanisms are defective if they are. So the DNA damage is produced by UV radiation to form thymidine dimers, but they cannot be corrected because there is a defect in the repair system. The results form into inborn deficiency of the enzyme nicking endonuclease. So this enzyme is deficient in those babies. And the next is like how they look like. That means increased cutaneous sensitivity to UV rays, produces blisters on the skin and dark keratosis and hyperpigmentation and atrophy of the skin and produce corneal ulcers. Then ataxia. So it's a familial disorder. So it is autosomal recessive and it has increased sensitivity to X-rays and UV rays. So here progressive cerebral ataxia takes place. Oculocutaneous telangiectacy. So it usually causes pulmonary infection, and lymphoreticular neoplasms are also a common condition. And IgE, which are antibodies, they are also deficient in 67% of the cases. The next is Bloom's syndrome, where the chromosomal breaks and rearrangements are seen in this condition. So the genetic defects are because of defect in DNA ligase. And here facial erythema and that is erythema swelling and redness, facial redness and photosensitivity. So they will be very sensitive to light. And then Fanconi's anemia. So this is autosomal recessive anemia because of defective gene in chromosome 20q and 9q. So this is a defective repair mechanism of cross-linking damage. So here an increased frequency of cancer by chromosomal instability is possible. Then hereditary non-polyopsis colon cancer. So it's inherited cancer. So faulty mismatch repair is seen here. And genetic defect has been located in the chromosome 2. And the gene is HMSH2. So this mutation in this gene can account for 50 to 60% of these cases. The next is recombination. So the genetic diversity, because if you see different organisms are different in their appearance or their characteristics. So that is because of the recombination and mutation. So mutations are not always dangerous. So only sometimes they are dangerous. So because of mutation and recombination, we will get different types of proteins and we will express different characteristics. Hence, we will be distinct. So the genetic diversity is maintained. So the recombination is nothing but combining the two segments of the chromosomes. So it's exchange of genetic material. There are two types of recombination. One is homologous recombination, which happens during meiosis, and second is transposition. So it is mainly by breaking and joining of the DNA strands. So here you can see the red segment and yellow segment. These two are exchanged. Okay, the bottom will get exchanged to the top strand. So the homologous recombination is nothing but, so this is involved in replication and cell division. So it is involved in the repair of replication forks. So recombination occurs with the highest frequency during meiosis, and the process which meiosis means in the germline cells, the replication takes place, the cell division takes place, and we get haploid gametes like sperm or ova. So here you can see the homologous chromosome pairing where the recombination takes place. So here you can see here finally in the red chromosomes you have some black edges. So that means the black, and in the black chromosome you have red segments. So that means there is a recombination that happened. So same thing. So this is called as the junction, which it forms is called as a holiday junction model for homologous recombination. So this is given in the name of scientists. Then transposition is nothing but it involves the movement of specific pieces of DNA in the genome, which are called transposons or transposable elements, also called as jumping genes. And so the type of transposition are two types: DNA transposition and retrotransposition. So in the DNA transposition, these transposons are capable of direct transposition from DNA to DNA. And this occur either by replicative transposition or conservative transposition. That means during replication or conservation. And it is common than the retrotransposition. But in prokaryotes, retrotransposition are easily seen. So transposition is nothing but it is a transfer of one segment from one part of the chromosome to the other. Okay, from somewhere it comes and inserts into the DNA strands. The retrotransposition, this is RNA intermediate is present. So here a copy of RNA is formed from the resposon, which is called retrotransposon, and here an enzyme reverse transcriptase plays an important role where DNA is copied from the RNA. So RNA acts as a template for the synthesis of DNA, and the newly formed DNA, this jumps from one segment to the another part of the genome and integrates randomly in somewhere in the chromosome. So like this. So this DNA goes and binds and integrates somewhere in the genome. So this is all about the DNA replication, repair, damage, and mutations and their positive, like if there are any defects. So what are the diseases they are caused? So do you have any doubts? Okay, so we will continue the next topic in the next class tomorrow. If you don't have any doubts, so I am ending the class. Thank you.